Anyway, welcome, Nora.
Well, Chris, thanks a lot. I want to thank Chris and Anto Bonci and Jonathan Pollock for their effort in organizing this course. And it's one of those things that I said I wish I had the time to stay here and listen to the talks and then just actually wander around this beautiful campus and interacting with everyone. But one of the problems that occurs once you start to get into professional life and administrative life, time gets out of your hands. So that's the only bad part of this. Now Chris and Anto said, Nora, can you give 2 talks? And I said, how am I going to say no to them? And so here I am, says, okay, I want to give 2 talks. And then and 2 hours, and I said, well, they wanted one of the, and I may have misunderstood whatever. So clearly one of them was going to be on science, and I hate to give administrative talks, but they wanted a talk that in terms of priorities of NIDA, but it's a priorities on the science ground. So that way, so I'm dividing my presentation in 2, and one of them is in research that's very much one of the areas that I myself as a scientist have devoted basically most of my professional life. And the other one is on the priorities, scientific for NIDA and great opportunities. So all of them are relevant to drug addiction, obviously, but rather than presenting you, I decided that my scientific talk, rather than presenting to you one that relates to, our studies to actually understand precisely how to, I'm trying to put this one in slideshow, how to understand the addiction in the human brain. I decided to do it to give you a perspective of the power of molecular imaging, because I was inspired by the brain initiative. What have I done? Yes, now the up or forward. I thought that I had a laser. Here is my laser. Okay, technology. I was inspired by the brain initiative and give you a perspective about the power of molecular imaging. And then I thought I should have also introduced findings for how powerful MRI is, but I was hoping that someone else may have. But if you have questions at the end, I can bring this up because it's also an extraordinary technology. But I was very intrigued about presenting to you molecular imaging because it is very, very relevant to what we do in terms of understanding the effects of drugs in the brain, and ultimately, at the end of the day, not just understanding what addiction is, but the toxic effects of the drugs, and very importantly, how do you translate that into treatment interventions, whether they are behavioral or medications. So for those of you that may not be aware of positron emission tomography, it's basically just an imaging technology that is based on the measurement of positron emitters. And positron emitters are radioisotopes that have the characteristics that they basically, when a positron is liberated, it leaves the place where it's liberated, and as it travels to tissue, it loses its energy. And when it loses its energy, it annihilates and generates 2 photons in the opposite direction. And so the PET technology takes advantage of this generation of 2 photons that go in opposite directions to actually quantify within a volume where that event took place. And so, for example, this is the crystal, the series of crystals that are in the detector that are surrounding the object. So when 2 photons arrive in opposite crystals at the same time, then you can actually know by probability that the event took within that volume of interest. And this property of taking advantage of these 2 photons diverting 180 degrees from one another is what really increases the sensitivity of PET technology over other nuclear medicine technologies like single photon emission computed tomography.
So is that a mirror image, or is it the same image?
No, this is what you're just looking at an event that disintegrates, and but when it disintegrates, it generates 2 photons in the same position. So you can actually literally determine that in that volume of interest where the event took place. And you are obtaining events in a circumferential fashion with multiple number of crystals. So then using back projections, you can generate the actual image. So it's not proper to speak about a mirror image, because what it really is a detection on an event by 2 crystals that are connected by the timing. as being able to determine where within a three-dimensional space where that happened. The other beauty about these positron emitters is that the isotopes are, you have carbon, you have oxygen, you have nitrogen. You don't have hydrogen, but you have fluorine, which actually chemically can behave very similar to hydrogen. And what it means is you can actually label compounds that are important for physiology. Most of the compounds you can label with carbon 11, without modifying its pharmacological structure. So, for example, I'm very interested in labeling cocaine. I can take one of the carbons from cocaine, substitute it for carbon 11, and then I have a compound that I can measure its concentration in the human brain, in the human body. Multiple applications. One of the most important has been in neuroscience, and another one as important has been in oncology, because by being able to label chemicals, you can see you can actually do analysis of the biochemical characteristics of a tumor without having to do a biopsy. But in the brain, it means you can look at biochemical parameters with actually non-invasively.
So what's the limit of the resolution?
The limit of the resolution actually, and again, if you look at it in terms of a physics definition, which enables you to quantify with precision what is the smallest element on your image where you can quantify the concentration, that's approximately 2 millimeters on the high resolution instruments. But you can visualize at a much higher resolution. But if you want to quantify in absolute terms, and I want to say, well, how many receptors are there, then your resolution is limited. But if you want to know where the receptors are located, not exactly how many, then your resolution is much greater. So it very much depends on the question. Well, one of the things that you know in what we have learned all along from initially experiments in animals and elegant work from multiple investigators is that all of the drugs of abuse that can produce addiction have this common characteristic of increasing dopamine in the nucleus accumbens, and that the mechanism by which they increase dopamine varies across the different types of drugs of abuse. And these mechanisms, in turn, while they have a common property because they are also divergent, explains why there are differences between the drugs, even though all of the drugs of abuse can produce addiction. And this divergence vis-a-vis the effects by which they increase dopamine also explains, too, why there are differences vis-a-vis other pharmacological actions of these drugs, like, for example, nicotine or amphetamine are drugs that can enhance attention, whereas heroin does not. But it also explains why there are different toxicities for these different types of drugs. Now, with PET technology, it is now possible to measure what you were doing in animals with microdialysis or voltammetry. Microdialysis, you insert a probe into the nucleus accumbens, and then you measure the changes in dopamine. Voltammetry, you also insert a probe. Now, one of the problems of inserting a probe, which is something that we need to address, is that that is an invasive procedure. And to start with, that's not going to ever work in a human subject. But even if we speak in animals, I think that one of the challenges that we currently have is inserting a probe will produce damage as you go through the tissue. So technologically, as we're thinking ahead, is can we start to extract some of these, can we develop similar methods where we need not to put any type of probe? With PET technology, the way that we're measuring changes in dopamine is indirect. And this is actually takes one of the methods, basically, or are similar. What varies a little bit is the specific radiotracer that you use. The one that's most widely utilized is raclopride. It's labeled with carbon 11. Carbon 11 has a half-life of 20 minutes, which basically decreases the amount of dosimetry, because it's radioactive material, into the subjects. It makes it very benign. And the beauty of carbon 11 is because basically it only has 20 minutes. You could do repeated studies in the same subject throughout the day, every 2 hours. So the way that we work it is C11 raclopride has a relatively low affinity for dopamine D2 and D3 receptors. And that means that raclopride can only bind when those receptors are not occupied by dopamine. Under normal baseline conditions, and these studies have been quantified in animals and in humans, your approximately 85 to 90% of your dopamine D2 receptors are not bound by dopamine. In other words, dopamine is binding to 10 to 15% of those receptors. So if you inject C11 raclopride, raclopride can bind to most of those receptors which are unoccupied. and you get an image that looks like this. However, if you bring the subjects back, which is the way that these studies are performed, on a different day, and you inject them with a drug, cocaine, methylphenidate, heroin, nicotine, alcohol, whichever drug you want to give, or with a behavioral intervention that could change dopamine, then you're going to change that availability of the receptors. And this is illustrated with methylphenidate, which is the drug that we have chosen to use because methylphenidate, like cocaine, its pharmacological action is to block the dopamine transporters, as well as the norepinephrine transporters. By blocking the dopamine transporters, cocaine and methylphenidate increase the amount of dopamine in the synapse and in the extrasynaptic space. Dopamine then binds to these receptors, and this is believed to underlie the rewarding effects of cocaine. Well, methylphenidate also binds to those transporters, increases dopamine, occupies the receptors. You give raclopride, and those receptors are occupied, so raclopride can no longer bind. And so what you actually do modeling to take into account the effects of your intervention in non-specific binding, in metabolism of the radiotracer. So after you do appropriate mathematical modeling, by comparing the availability of receptors under this condition versus this one, you get an estimate of the relative changes in dopamine. And these relative changes in dopamine have been shown to be linearly related to the changes in dopamine that you obtain when you're doing microdialysis experiments. Now, we and others have used this technology with some modifications. We use methylphenidate. Many other people have used many other drugs. But we have used methylphenidate. We've given it intravenously. We've given it orally. Methylphenidate is a very interesting stimulant drug, very similar to cocaine pharmacologically. Animals don't distinguish intravenous cocaine from intravenous methylphenidate. We've given intravenous methylphenidate to multiple cocaine abusers. They don't distinguish it from intravenous cocaine. Now, on the other hand, methylphenidate is Ritalin, which is the psychoactive drug that's most frequently prescribed in children in the United States, which is given to the treatment of attention deficit hyperactivity disorder. So we have the same drug that can have very different effects when you give it intravenously than you give it orally. Intravenously, it's very reinforcing, very rewarding. It produces an intense high. It can produce addiction. People become addicted to when they inject methylphenidate, they can become addicted to the drug. When you give it orally, some people do become addicted, but it's a much rarer variant and usually occurs with huge doses when people start to take very large doses. So here you have intravenous methylphenidate, blocks the transporters, dopamine then occupies the receptors, raclopride binding goes down. And in these individuals in which we are giving intravenous methylphenidate, we also can assess the behavioral effects, which is a beauty of doing studies in humans because you can actually directly probe the subjects in whom you are making the measurements. And in this case, for example, this study, we gave them intravenous methylphenidate, a heptic dose, 0.5 milligrams per kilogram IV. And then 1 minute later, we injected raclopride and then do the measurements of the changes in the specific binding here, which basically is directly reflecting relative changes in dopamine. And what we found is we published a long time ago was that a very strong correlation between the changes in dopamine produced by methylphenidate and the subjective perception of high of individuals. Those subjects in whom methylphenidate produced very large changes in dopamine, again, these are relative changes, have very intense high. Whereas those subjects in whom this dose of methylphenidate had no observable effect, they didn't feel anything, we couldn't even detect cardiovascular responses, even though we were controlling for plasma concentrations of methylphenidate, did not report a high. Indicating that in humans, the ability of these drugs to increase dopamine is associated certainly with a subjective experience of the reinforcing effects of the drug. Similar results have been reported by the group at Yale using intravenous amphetamine, and many investigators since then have applied it and have shown exactly the same finding, whether it is alcohol, whether it is nicotine, whether it is cocaine. The larger the changes in dopamine in human subjects produced by these drugs, the more intense the experience of high, euphoria, well-being.
So that isn't because you metabolize it differently?
No, it's not. We counted. That was an important. We actually control for two things. We control for the concentration of methylphenidate in plasma, And this is basically does not in any way relate to the concentration of methylphenidate in plasma. But we went beyond that, and we actually quantified the number of dopamine transporters that were blocked by methylphenidate. So just like I'm showing you that you can look at receptors, you can look at transporters with ligands that bind to dopamine transporters. So we label C11 methylphenidate. We've labeled C11 cocaine. And those molecules bind to the dopamine transporter. So you can use it as markers of dopamine transporter availability. So this is not accounted by differences in blockade. This is not accounted by differences in plasma. This is accounted evidently by differences in the ability of the dopamine cells to release dopamine. So that is what's accounting for this very large variability between subjects in the response to this drug of methylphenidate at this dose. Now, later, one of the things that generated a lot of attention and people were very upset, the psychiatrists were very upset at me because when we first started to work with methylphenidate and we were comparing side by side intravenous methylphenidate, intravenous cocaine, labeling it with positron emitters, measuring changes in dopamine. So the psychiatrists were very offended when I published a paper that I think it was not politically correct, but I did, which was called something is methylphenidate like cocaine or something like that in the main journal of psychiatry. So people got very offended with that, and they said, well, we are giving this drug to children. Well, and then they said, well, why are we not seeing more addiction? And one of the concepts was then they were saying, well, when you are giving oral methylphenidate, you are not changing dopamine. And I said, well, what is the logic, the same drug? Well, the logic is very real. When you take a drug orally, recall, it has to pass through the gastrointestinal tract and it goes to the hepatic circulation. It goes to the liver. And there it gets metabolized. And in the case of methylphenidate, methylphenidate is metabolized into ritalinic acid. And ritalinic acid has very poor stimulant properties. So one of the hypotheses in the clinical world was that oral methylphenidate was not rewarding because it was widely metabolized to ritalinic acid. So we decided to do the experiment, and we did, okay, instead of giving intravenous methylphenidate, let's give oral methylphenidate. And we did the experiment. Now, when you take a pill orally, guess what happens? When you inject and the thing goes immediately from here into your heart, and then it goes from the left heart into the lungs, and then into the right, and then it goes to the brain. When you take it orally, and so this process of injecting, we've actually estimated that when you inject intravenously, you start to see the presence of the drug something like 45 seconds in the human brain. If you take it orally, it has to get absorbed, it goes this, and it's a very, very, very slow process. So we could not inject in intravenous methylphenidate. We could do 1 minute later raclopride. But with oral methylphenidate, we couldn't, because if we do it 1 minute after we give it orally, you'll see nothing. So we had to wait 60 minutes, because that's when you start to see behaviorally some of the peak effects of oral methylphenidate. And what we found was very intriguing, that we saw very significant changes in dopamine. So the whole hypothesis that oral methylphenidate is not a stimulant drug because it's metabolized actually into ritalinic acid is not correct. Oral methylphenidate at the doses that we use to treat ADHD is a potent stimulant that actually produces significant changes in dopamine. But what's very interesting is that you are not producing a high. So whereas in this case we see this very nice regression, and by the way, if I am impressing you with the beauty of this regression, I'm also going to confess that I've never ever since then gotten a regression that is as strong as this one. This was 1999, and we're always looking at association between behavioral and biochemical variables. This is the best regression we've ever had in our lives. So when we give it orally and there was no association, so which brings to light, what does it mean? Now, this could not be bogus, plus it has been replicated many times. So then what does it tell us? It tells us that dopamine may be very important for reward, as have been shown by the microdialysis experiments or voltammetry. But it also tells us that it's not just the amount of dopamine, that there's something in their way that dopamine is changed that is influencing whether you are getting a high or not. Now, you go when you're facing data, you of course try to understand, well, what have other thoughts about it, because many bright people have been thinking about these things. And there's an investigator that died a couple of years ago, Schuster, we got working with another investigator who is still one of NIDA grantees, Bolster, who had done an elegant, simple experiment. And he actually they were working with monkeys, and they were studying cocaine, and they wanted to know how does the speed at which the drug gets into the brain affects its rewarding effects. And they did a very clever experiment. They injected the animals. But what they varied was the rate at which they injected. So some of the monkeys were injected very, very fast, whereas in the other ones, the injection of cocaine was very, very slow. And what they reported in this paper was that when you injected very rapidly, the effects of cocaine were much more rewarding. Actually, that the consequences of injecting rapidly was basically accounted almost as if you were giving double that dose when you injected slowly. And they were the ones that unequivocally show that the rate at which these drugs are entering the brain will determine its rewarding effects. And I think that's exactly what we're observing here. Here we have 60 minutes, so if you're speaking about slow, 60 minutes is extraordinarily slow. Here we're speaking about very, very fast. And I always confess this also. I mean, for years, it's embarrassing, but for years, I was trying to say, well, why the hell? What is the mechanism of the rate dependency? And I feel very dumb because it's very obvious. I mean, it's very obvious now, but it was not obvious to me. And my brain sometimes gets very convoluted. So I was one day in the kitchen thinking, and then all of a sudden, I realized that the refrigerator was making noise. But I realized that the refrigerator was making noise because it stopped making noise. So then, of course, as a neuroscientist, I went back and I started to read about these auditory cortical neurons, and I realized that there are cortical neurons that respond when there is a delta function. So they don't respond per se to the intensity, but when there is a discontinuity in the signaling, that's what activates them. And I say, ha, there it is. That's probably why it is reward. That there must be cells that get activated when there's a discontinuity in dopamine. So there's an abrupt increase in dopamine when you inject. And then one day it just hit me and says, no. Stop this convoluted thinking. It's very obvious. And this is what's very obvious. Look at physiology. The dopamine system, again, learned from nature because nature has taken millions of years for evolutions. The dopamine cells fire in two modes of operation. And those modes of operation signaling and have downstream consequences that are very, very different. So you have phasic dopamine cell firing, you have tonic dopamine cell firing. When you have phasic dopamine cell firing, you have these trains that are very short lasting, but very, very fast firing that leads to very abrupt increases in dopamine. Tonic dopamine cell firing, on the other hand, is a slow firing with relatively low release of dopamine, but it's constant. Both of these dopaminergic signaling are very, very relevant on how this neurotransmitter works. The tonic dopamine cell signaling is enabling you the readiness for responsiveness. But this phasic dopamine cell firing, actually, when that happens, it signals a saliency response in your brain. Now, what are the consequences at the molecular level? Well, this one is going to produce very large changes in dopamine that are going to be able to stimulate receptors with low affinity. So we know that the dopamine D1 receptors, for example, have lower affinity. So when you have phasic dopamine cell signaling, you are going to be activating low affinity receptors as well as, guess what? With the high affinity. So when you're doing phasic dopamine cell firing, you're going to be activating dopamine D1 and dopamine D2 receptors. When you do tonic dopamine cell signaling, which actually leads to much lower dopamine concentrations, you're going to be predominantly stimulating dopamine D2 receptors. And now we know, for example, from animal experiments, that it is really the optimal stimulation from cocaine occurs when you are stimulating both dopamine D1 and dopamine D2 receptor signal. And so it finally just clicked in my brain, something that was so very obvious. The reason why drugs are rewarding is not just that they are increasing dopamine, but that they are emulating the levels of dopamine that are produced by phasic dopamine cell signaling, which is the mechanism by which the dopamine neurotransmission not only signals reward, but it also triggers conditioning. And as you know, conditioning is at the essence of the process of drug addiction and all of the neuroplastic changes that you are observing. So this is one of the examples about how through molecular imaging, by looking at the kinetic process of the behavior of these drugs and the changes in dopamine, you can actually go beyond understanding the role of a neurotransmitters in terms of concentrations, but its dynamic effects. Now, with molecular imaging, you can also probe the question, well, you are producing these very fast changes in dopamine when you are injecting cocaine or you are taking methamphetamine or heroin. But what happens when you do it repeatedly? We know that biological systems, basically, and we know that from electrophysiology in the brain, we know that for actually almost any organ system, that if you perturbate it very dramatically, then you initiate homeostatic changes that are going to try to basically buffer that suprastimulation. So the question that then follows is, if you're giving and giving and giving this supraphysiological stimulation with dopamine, what happens to the dopaminergic system in terms of adapting to it? In other words, are there changes that occur in the function of the dopamine neurotransmission signaling with the repeated administrations of drugs in human subjects? So we have been doing for many years, many studies to try to answer this question, and many other investigators have been doing now so too, and many animal studies. And one of the most consistent findings that actually I want just to simplify is that what we have observed and what others have observed with imaging as well as with postmortem studies is that in people that are addicted to drugs, one of the consistent findings is that there is a reduction in the expression levels of dopamine D2 receptors. And similarly, in studies in laboratory animals, in rodents, for example, that are prone to self-administer drugs, they tend to have much lower expression of dopamine D2 receptors than those that do not. And this is just simplifies, summarizes some of the studies that we have done in cocaine abusers, methamphetamine abusers, alcoholics, heroin abusers, and others have done nicotine abusers, and across all of the addictions, except one. But I'm not going to tell you unless you ask me, but except for one. All of them, you see a reduction in dopamine D2 receptors. Yes, and you can ask me at the end of the day, but it's one of the ones that, I mean, I guess there's always an exception in life. No matter, I basically look at this data, we're not seeing a decrease in dopamine D2 receptors in marijuana abusers.
Or did you get a reduction with oral amphetamine? Oral Ritalin?
Cannabinoid? With oral Ritalin, in terms, yes, absolutely. We did a study where we actually, was one of those most complicated study, that very complex study, not complicated, complex. We took people with ADHD, had never been treated, measured their dopamine D2 receptors, studied them for 1 year where they are consistently being treated with methylphenidate Concerta, retested them, and see a downregulation of D2 receptors. At the same time, we are following a group of controls, no treatment, we retest them, no changes in D2 receptors. So they downregulate with chronic methylphenidate. After 1 year, they downregulate.
But is there any baseline difference between people with ADHD?
In D2 receptors, no.
So it's not D2.
No, we did not see a difference in dopamine D2 receptors in individuals with ADHD that have never been treated. We see a difference in the magnitude of changes in dopamine produced by a challenge. So it's attenuated. And that gets further attenuated when you treat them 1 year with methylphenidate, as if they were developing a tolerance to the drug. Now, what we didn't do was follow them to try to see when they downregulate, how long is that downregulation lasting? That we did not. We haven't. We just did 1 point.
In your first study there in 1999, were those drug-naive patients?
Drug-naive. That first study was done in drug-naive patients. We've then replicated it in individuals that have been exposed to drugs, and now what we do is, as you may imagine, intravenous methylphenidate is quite a potent stimulant, so we don't want to give it to someone that is naive. So we basically include subjects that have had experience with cocaine in the past or something, but they are not abusing it.
What about people, like, for instance, with ADHD that have been treated with prescription stimulants? It would, in theory, affect their tolerance for methylphenidate.
You should.
Right.
Yeah, but we haven't done that. We haven't taken, except in this particular study where we treated them 1 year, okay, so they've been treated 1 year, then we give them IV methylphenidate, and that is attenuated, that's blunted. it's blunted. And we did the same experiment in the controls to just be sure, because the whole notion of sensitization, and there was a paper on the archives that showed sensitization in humans, so we needed to control for that. And we did exactly the same experiment, except we didn't give them Concerta, retest them, and we do not see neither sensitization nor tolerance.
I was just going to ask, with the drug-abusing subjects, do you think they've got the lower D2 to begin with, or do you think it's a result of the drugs, or do you think there's a combination of the two?
I think that, I mean, you're giving me the easy way out. When I always say to people, when you're in doubt, say the combination of the two. But I'm not saying it facetiously. I think that in the process, obviously, we and others have been doing a lot of work, and there is evidence, as I was commenting, that in rats, strains of rats like the Fisher and Lewis that differ in the administration, also differ in the levels of dopamine D2 receptors. So there are significant differences, as we know, in strains of animals on the levels of D2 receptors in the striatum. Among humans that don't abuse, and you see it here. So here I have two different studies. These are inpatient studies and cocaine abusers, but the cocaine abusers are inpatients. I studied them with another radioligand, N-methylspiroperidol. And these are outpatients, cocaine abusers in green and pink, normal control studies with raclopride as a function of age. So this is the measurement of receptor availability. And what you see that cocaine abusers are lower. But look at the normal controls and look at the distributions. So you have this subject here that has much higher levels than this one here. In fact, this looks like a drug addict in terms of the levels. So there is tremendous diversity in the levels of D2 receptors. And we are very interested in understanding what accounts for the difference in the expression patterns of dopamine D2 receptors. Again, if you want an easy solution, genetics, right? I mean, sort of says your genes are going to determine the expression levels. But I'll get to that sometimes that response is not so straightforward. But there is some data from, and actually we've been looking at polymorphism on the D2 receptor gene to try to see if it influences D2 receptors. And you can say, I don't know. And some investigators say yes, and some investigators say no. We've started, one of our colleagues, Selina Shumai, at the laboratory, started to work with the AKT polymorphism, which actually is involved in the internalization signaling for the D2 receptor. And that we show has a very significant effect. It's the strongest effect we've had with D2 receptors. So clearly genetics are involved, but clearly the environment. And also, like he was asking me, those exposure to stimulants downregulate. We've shown that stimulants for therapeutic purposes downregulate. So just like in animals, exposure to cocaine downregulates D2 receptors. That's why I say, I don't say it facetiously. It's a combination of both. And an intriguing combination, because what is emerging, I think that, and I don't know if I have the slide or not, because I actually was trying to give a talk on molecular imaging, not just per se on the process of addiction, is now the consensus is that having low levels of dopamine D2 receptors in the striatum creates a vulnerability factor. And I hope I have the slide that I would like to show you now. Here it is actually demonstrates that we've done 3 different experiments, but and what our experiment is manipulating the expression levels, increasing the expression levels. So the question is, if we can increase the expression levels here, and we're speaking about having low levels of D2 receptors as a factor that makes you vulnerable, then we should be able to help. It's not addiction because you see overlap, but it's a vulnerability factor. And other investigators, like Eric Nestler, has been basically producing manipulations to actually interfere with the function of the D2 receptor intracellular signaling cascade. What we've done is overexpress D2 receptors in animal models of consumption of high doses of drugs and inquire then what happens to consumption of drugs. And what happens is very interesting, because I think it's extremely telling of why low D2 receptors are actually making you vulnerable. So this is the first study. This was we did a long time ago, 2001. Peter Tanos, who was a postdoc, had just come as a postdoc on the laboratory. And in this first study was in alcohol, Sprague-Dawley rats. They are made to self-administer alcohol. High doses, when they are administering at high doses, the animals are injected with an adenovirus inside of which is the D2 receptor gene, stereotactically into the nucleus accumbens. A control group is injected with the adenovirus, but with no D2 receptor gene for controlling. And when you do that, and you can measure then, of course, what happens, percent change in D2 receptors in the nucleus accumbens, these are days after injection, so this is the first vector. At day 4, you have the maximal expression, and the levels of receptors have increased approximately 50 to 60%, and then by day 10, they are back to baseline. You can inject the animals again, day 20, and the receptors go up again. And so within these conditions where the animals are overexpressing D2 receptors, you ask the question, well, what's happened to the behavior of administration of alcohol? So we've done alcohol, cocaine, and we've done alcohol also in preferring and non-preferring rats. The findings are the same. So I take this first paper. It's exactly the same. What happens when you overexpress D2 receptors is this is percent change in alcohol intake. So you produce a dramatic reduction in the amount of drugs consumed. And that's the same story with cocaine. It's not that you block it. The animals still administering some alcohol, or the animals still administer some cocaine, but they are administered relatively very low doses. And as the receptors go back to baseline, you can see that the drinking behavior goes back to baseline, then starts to lose its significance. You inject again, and you see this abrupt decrease in the amount drunk. These are the animals that are the control animals. There's no effect in terms of their drinking behavior. Exactly the same thing with cocaine. And what to me is telling is that what it seems to be doing, overexpressing D2 receptor, is interfering against the administration of high doses. High doses of alcohol, high doses of cocaine. And what is alcoholism? The consumption of 4 drinks if you're a female on one sitting, or 5 drinks if you're a male. It's the consumption of high doses. How does it manifest if you have not seen a person that's addicted? A person that's addicted assume this is alcohol, I'm an alcoholic, I'll take it, that's not sufficient. I have to get more and more and more, I become compulsive. And it is a compulsive pattern. And if you've seen it in the movies, if you have not seen it in reality, if you're lucky enough to have not encountered someone that's close to you that's an addict, you'll see it. It's basically almost like insatiable thirst. More and more and more and more. And that compulsive, it's almost like a repetitive behavior. It's almost like a perseverative behavior. And if you ask, and I've asked, because I'm a very curious person, so and also as a psychiatrist, I ask questions that people feel embarrassed to ask, but I ask them. So one of the questions that I do ask patients is, why are you taking cocaine? Is it so wonderful? Yeah, or alcohol, or heroin, or methamphetamine? And what I get is, you get all sorts of explanations, but one of the ones that's very intriguing is, because I get it many, many times, is, I don't know why I'm taking. I don't know why I'm taking. It's no longer pleasurable. I just cannot control it. So there is that description, spontaneous description of lack of control. And that, since, I've been dwelling on that to me, and as a psychiatrist, and my brain is the most associative brain. It travels from here and there and gathers information, so it's not voluntary, it just happens. It basically, that immediately reminds me of patients with obsessive compulsive disorders. I don't know if you've seen them, but actually they come to you and they want to be treated and their hands are bleeding. And so they wash and wash and wash and wash. They don't want to wash their hands anymore. They know they're clean. They just cannot control it. And so that sense of loss of control that drives the compulsive pattern of behavior is in part stamped by the low levels of dopamine D2 receptors in the striatum. And now investigators have actually shown that to be the case. The group of Trevor Robbins has shown, for example, that animals with low levels of dopamine D2 receptors predicts those animals to be impulsive and to have the propensity for compulsive administration of cocaine.
Nora?
Yeah.
Actually, what's been shown also in rats is that the decreased D2 binding in the ventral striatum is first restricted to the shell, and second is also attributed to decrease in autoreceptors, presynaptic receptors. So when you play with the D2 in the striatum, does your virus cross the synapse, and do you also play with the presynaptic ones?
Unfortunately, it's something that we've always salivated at the opportunity to do. But with PET technology, we cannot differentiate a presynaptic from a postsynaptic.
But with your manipulation on rats?
Yeah, no, with manipulations on rat, absolutely. And one of the scientists at Brookhaven National Laboratory has started to work with these knockouts that basically have the knockout on the presynaptic versus the postsynaptic D2 receptor, to start by looking how much of the signaling on raclopride is related to presynaptic D2 receptors, because they have been dismissed. And on these preliminary results that she has in mice with C11 raclopride, approximately it appears that 30% of the signal comes from autoreceptors.
Actually, and we got the same with in situ organization paper that just came out in high impulsive animals.
So 30%.
Yeah.
Yeah, so that's,
So that's a little difficult to do because you're taking away the break from dopamine release, right? So then you can occupy more of the.
correct.
se-
Absolutely
and then
correct.
you're gonna
Absolutely.
internalize those and get rid of them. So it's
No,
a bit
absolutely.
tricky.
No, it is tricky. It's a very indirect, and that is a major confound. It's a major confound because you are going to release more dopamine. That's going to decrease your binding. Absolutely. So it is, but we don't have. I think that actually we, of course, and a lot of people have started to think, are there certain ligands that could predominantly bind to autoreceptors, and as of now, we have not been clever enough to come up with a strategy. But it would be extremely relevant because obviously, and as you know, on all the transgenic mice, that they're modifying the function of the autoreceptor, that in turn modifies the responses to cocaine. And we really do not, what we can do, and when we're doing these viral injections, we actually, those viral injections that we've done in the past are not specific. I don't know where the D2 receptor is going. Now, with the technologies that are much more advanced, we can selectively have the D2 receptor expressed postsynaptically versus the D2 receptor expressed presynaptically. Moreover, we can actually inhibit the expression postsynaptical or presynaptically. And from the animal perspective, that is one of the ways of going about it. Of course, there are other ways, but for a long term, that's a way of trying to address it. Now, we have been trying to, so if you have low D2 receptors in the striatum, and again, I do not know with the, because of the simplicity of thinking, we have been thinking that a predominant signaling is postsynaptic, but I cannot, basically, I do not know that it's necessarily just postsynaptic. It could be D2 receptors sitting in the glutamatergic terminals that come from the cortex. We do not know. They are down, and that is associated with these compulsive patterns of administration. Now, in the human subjects, we've been able to, at the same time that we're measuring D2 receptors in their brains, we also measure brain glucose metabolism. And we measure brain glucose metabolism because this is a very good marker of brain activity. And as a result, it's one of the radiotracers that used extensively to make early diagnosis of neurological diseases. So, for example, you can make a diagnosis of Alzheimer's disease before the patient actually develops the symptoms. And you can do that initially. They were saying, well, you look at decreases in metabolism in the hippocampus before the patient actually develops symptoms that are serious enough to attract the attention of the family or the clinician. And now if you want to even go earlier, you see decreases in metabolism in the precuneus. The precuneus is an area in the posterior parietal cortex that is probably the most interconnected area of the human brain that we know of. So it's the main hub of one of the main hubs of the human brain. And you see decreases in metabolism in that area very, very early. Similarly with Huntington chorea, you can see that decreases in metabolism in the caudate before the symptoms emerge. And that gives you an idea. When you start to see the decreases in the caudate, you know that the symptoms will start to emerge. So we use it as a marker of brain activity. And we measure in the subjects D2 receptors, so we've done it in cocaine abusers, 2 different groups of cocaine abusers, 1 group of methamphetamine abusers, 2 groups of alcoholics. And we've replicated these low receptors across all of them, but we've also, in looking at brain glucose metabolism, we've also seen this replication. One, first of all, when you study people that are detoxified, that have been, that are not taking actively cocaine, but are in the hospital, they have been in the hospital for at least 2 weeks, which is what we at least have them, ideally 3 weeks. What we've been reporting for years now is that they have a very significant reduction in metabolic activity in prefrontal areas. And while there are several frontal cortical regions that are affected, as well as some of the temporal areas, the most affected one are the dorsal anterior cingulate gyrus, as well as the ventral part of the ACC, Broadband Area 25, as well as orbital frontal cortex, both the medial and the lateral. And those are very, very significantly attenuated, whether you're a marijuana abuser, whether you're a cocaine abuser, whether you're an alcoholic. So these findings are consistent across different types of drug addictions. And because the same measurements are in both groups of subjects, we've actually assessed, this is for cocaine abusers, this is for methamphetamine abusers, this is for alcoholics, what's the relationship between dopamine D2 receptors and, in this case, brain glucose metabolism in the orbital frontal cortex. And the same finding is for single-age gyros. The lower the receptors, the lower the activity in this brain region. The higher the receptors, the higher the activity in this brain region, indicating to us, certainly, that one of the mechanisms that is responsible for why having low levels of dopamine D2 receptors is promoting this compulsive phenotype that activated my dopamine cells.
Summer rain, look at that.
I don't think my flight is leaving.
I would go by train if I was you.
What's your flight?
Not stay.
What's your flight number? I can check.
It's a 6 o'clock flight, LaGuardia,
With what airline?
US Air.
US Air.
The weather channel says 6:30 thunderstorms.
So otherwise I'll need a room, otherwise I'm homeless.
There's
Follow the
plenty of
discussion, there's plenty of questions.
Yeah, yeah, no, but the bottom line is, though, so why is it that one can say low levels of dopamine D2 receptors, this association with these brain regions, is if you damage these regions in animals, or if you have damage of these regions because you have a meningioma in the orbital frontal cortex, or you have a stroke, or you get hit with a bat in the orbital frontal cortex. There's the paper on Science of that guy that got the train rail through the orbital frontal cortex, the Kineos gauge, that led to these repetitive behaviors. What you do is repetitive behaviors, and it's a pattern. It's almost like they just repeat themselves. They don't have a purpose. And yet the animals who have this damage, or the humans who have this damage, just basically follow them, and this in a human can be very devastating, and in an animal it generates a pattern of behavior that is very irrational, apparently irrational, because the animal will press a lever when it's no longer rewarding, or they will drink water and water and water even though they are no longer thirsty. They lose the flexibility to change the behavior as a function of the reinforcing characteristics that are present in the context, which are very necessary, right? So if I didn't choose the motivation to drink this because I already am no longer thirsty, and I keep on drinking and drinking and drinking and drinking, if you damage my orbital frontal cortex, probably that will happen. So I lose the capacity from a reinforcer that's valuable. Once I drink it, is no longer salient, and I can go and do something else, to that incapacity. So that, in turn, triggers me repeating the behavior. And this is likely to be one of the reasons why low levels of dopamine D2 receptors may be generating this phenotype. And I want to show you actually a slide that shows this even at another more profound level. Yeah.
Is it also the case in OCD patients, like the brain metabolism and the D2 receptor, like are they also lower?
In the OCD patients, the dopamine D2 receptors are not the main culprit that you observe. It's the serotonergic system that is profoundly disrupted. So I've always been very intrigued about the notion of the belief that if you enhance serotonergic signaling, because the orbital frontal cortex is widely innervated by serotonin fibers, you may be able to help buffer that compulsive pattern. And if you look at the way that clinicians, it's not an approved medication, treat patients with cocaine addiction, many of them use SSRIs, serotonin reuptake inhibitors. NIDA funded a series of studies, randomized clinical trials, that had to be terminated because the clinicians could not increase the dose sufficiently. They start to have untoward cardiovascular effects. And so there hasn't really been a good clinical trial high enough doses to document it, but clinicians are using it. So again, what happens is, you have redundancy of neurotransmitter systems that are modulating actions in the brain. And in the case of obsessive compulsive disorder, you have abnormalities in caudate, you have abnormalities also in the dorsal cingulate, and you have abnormalities in the orbital frontal cortex. But there is a predominant serotonergic disruption. Interestingly, if you have a patient with OCD and they take cocaine, their symptoms exacerbate. And I also had one of my patients that I did, one of the subjects that volunteered for the PET scan, which was, it got imprinted in my brain because it was the most unusual patient I have ever had. Was unusual, he was like 42, 43 years of age. He claimed he was taking something like 10 grams of cocaine a day, which is huge dose, injecting. The other very unusual thing about this case was that this patient had started to take cocaine 3 or 4 years before. Before that, he did not have a history of drug abuse. And this was unusual. I had never seen such a patient. Usually, you have someone in the forest, they have a long, long history of drug taking. The story of this patient was he had been referred to the psychiatric hospital, actually with a lawsuit pending from the American government because he was a gambler. And he had been a serious, serious gambler and owed the government something like $1 million. And he had tried all sorts of treatment interventions and nothing had worked. Until one day he tried cocaine. That cured his gambling. He stopped gambling, but his cocaine escalated. And so he had a severe problem with addiction. So again, the notion that this neurocircuitry, basically there is an overlap of these systems you can observe it in the presentation of these clinical cases. This is actually shows that relationship. So what you're seeing here is the statistical parametric mapping analysis that allows you to plow through the three-dimensional volume of the human brain and to inquire which areas of the brain, not the other studies were done pre-selected regions, prefrontal, blah, blah, blah. This one actually is not like that. This one you just ask the analytical tools to identify the areas where these two are correlated. D2 receptors in striatal brain glucose metabolism. And these are not addicted people. These are individuals who have a family history of alcoholism. And this is a study that I did with Henry Begleiter when he was alive. And they had a father who was, a biological father who was an alcoholic, and then they also have a first-degree relevant who was an alcoholic. But they themselves, they were adults, but they were not alcoholics. And we tested them with both of them, and these are the areas exactly with low levels of D2 receptors, or levels of D2 receptors were positively associated with glucose metabolism. So if you had high levels of D2 receptors in striatum, you have high metabolism in the orbital frontal cortex. This is medial, lateral, dorsal anterior cingulate cortex, dorsolateral prefrontal cortex. We also observe that now you're plowing through the whole brain in the anterior part of the insula and into the caudates, and these are the regression lines. And it is very interesting because it actually shows in these individuals at risk that are not taking drugs. I mean, what's very interesting too about this particular study was, again, it was a disaster for me initially as trying to get the grant because I couldn't get the grant, but actually the data was the data. What we found was that these subjects had higher levels of dopamine D2 receptors than those without a family history. And this was contrary to our expectations. But despite the fact that they have higher levels of dopamine D2 receptors, higher than those of non-family positive individuals that have histories of, no history of alcoholism, but no family history, is that their levels of metabolism in these brain regions did not differ. So in other words, they needed to have higher levels of D2 receptors for equivalent levels of brain glucose metabolism in these areas of the brain. And we then postulated the reasons why these individuals are not alcoholics is that the high levels of receptors in the striatum that they have is leading to actually proper normal function of these prefrontal areas. And I predict that if they were to start taking drugs, this would lead to a reduction on D2 receptors in the striatum that will destabilize the prefrontal cortical areas and result in the compulsive phenotype. So this is where we actually come through in using this molecular imaging to try to understand mechanisms that may drive some of the effects that relate to the addiction phenotype. Now, one of the things that as we're studying drugs of abuse, what you actually see, and in terms of what many times alerts our society to pay attention to the effects of drugs, are not necessarily per se the process of addiction, but in many instances what attracts them is the toxic consequences of the drug taking. And that is something, again, for which molecular imaging provides you a very sensitive indicator that opens up new mechanisms about how they be actually working. And I'm going to illustrate that with nicotine, cigarette smoking. Cigarette smoking is the main way by which people absorb nicotine. And we've done enormous amounts of studies throughout the years in neuroscience to actually investigate the effects of nicotine on nicotine receptors. And most of the work of nicotine is on nicotine receptors. But guess what? Nicotine acts well beyond just acting at nicotine receptors. And this has been noted by investigators. For example, there is some evidence that nicotine may have actually effects, we don't know if it's direct or indirect, on histone deacetylases. But there was a paper that was published many years ago in the 80s in a sort of a pretty bad journal. I shouldn't say bad journal, I said a journal with a very low impact. Yes, you forget, I didn't say bad journal. Guys, I apologize. I really thoroughly apologize. I published in that journal, but it's a very low impact journal. So, Life Sciences. And this was a fascinating... I apologize. I do like, I mean, there are some fantastic papers that have been published in Life Sciences, and this one was one of them, in which they had, no, I thought it was an amazing study where they had given nicotine to pregnant rats, and then they actually monitored the offsprings, and they observed that the offsprings of these rats that have, the offsprings from the mothers that have received nicotine during pregnancy have a much lower concentration of cells in the hypothalamus that express the enzyme aromatase. Now, why is this important and why should you care about it? Aromatase is a very important enzyme for males and females, but I would say about in fetal development, it's a particularly important enzyme for the male brain. Because aromatase, there are two factors that masculinize the male brain into being a male brain, which, by the way, is different than the female brain. I guess because I'm a female, I can say these things, right? I'm very intrigued about it.
It's kind of sexist, isn't it?
Yes, it is sexist. But something, some advantages give to the female species, right? I mean, there are some advantages that we can say these things and not feel constrained. But anyway, I apologize if someone, I mean, I should be more serious. I apologize.
You shouldn't.
No, it's all right. You're on tape.
It's on tape.
The world's going to hear you.
You're going to edit, right? You're going to edit my statement about life science.
You might want to just do a little bit of correction.
Yeah, the life sciences I want corrected because I do like it. No, I do like it. I don't want to offend anyone. So they show that, so why there are two processes by which you masculinize the male brain. One of them is, of course, the XY chromosomes. And that will basically result in different imprinting of genes. And it will also result in different expression of genes. And that is very important. But the other one is this enzyme aromatase. And it so happens that in fetal development, and I don't know exactly at what stage in humans, or I don't recall it in rats, there is a surge in testosterone. And that surge in testosterone actually results in an increase in concentration of estrogens. And this is mediated by aromatases, which then transform the testosterone into estrogen. If you inhibit this process, then you're going to be interfering with the masculinization of the brain as it relates to the process mediated by these aromatases. And of course, researchers have investigated to try to understand, well, for example, the masculinization through the chromosomes is related very much to sexual behaviors. So sexual preferences apparently are not mediated by this aromatase. But what appears to be mediated to this aromatase, not that it's the only one, but it's very significantly mediated, is territoriality and aggressiveness. So if you inhibit this aromatase, then you generate male offsprings that are going to be less territorial and less aggressive. On the other hand, in a female fetus, if you give testosterone, and that then gets transformed into estrogen, or you give them estrogen, you're going to actually make them more aggressive and territorial. So this has been observed in the rats. But this was reported in this paper, which I thought was quite fascinating, and nobody had actually followed it through. But it was nicotine. So the question was, does this happen in humans? Now think about it. I shouldn't say. Currently, approximately 17% of women in the United States that are pregnant smoke through pregnancy. So you're going to be delivering nicotine into the fetus. So the question is, are you going to be potentially inhibit this aromatization process? So that's the question. If you give nicotine at the concentrations that people achieve when they are smoking cigarettes, do you inhibit this particular enzyme? So at Brookhaven National Laboratory says, well, how do we actually get the measurement of the concentration of aromatase? So one of our chemists, Sonny Kim, went into the literature and said, well, there is a drug, Borosol, that is an aromatase inhibitor that is actually used for the treatment on patients with breast cancer, where you actually want to inhibit the production of estrogen. It's also used in certain male cancers, again, as I think it may be prostate. But so there is a drug, Borosol, and so he labeled the Borosol with a positron emitter, and here you sort of actually have it, carbon-11 Borosol, and these are images of a baboon, a non-human primate. It's a coronal image, and the uptake reflects where you have the highest concentrations of the enzymes, where you can see it's actually very high concentrations in the hypothalamus, but you also have very, very high concentrations in the amygdala. This animal is after administration of 0.015 milligrams per kilogram of nicotine, which is a very low dose, and you can already see a decrease in the binding. And this is a 0.3 milligrams per kilogram, which is actually a dose within that reaches plasma concentrations equivalent to those that smokers are achieving. So at the doses that people are smoking, you are actually in the monkeys inhibiting the concentration of this aromatase. And we just started the first human subjects, and this is an aged male non-smoker. This is a male smoker, so this non is a mistake. And this is, again, a patient who's being treated with the medication, the Borosol, which is the letrozole, which is the aromatase inhibitor. And you can see certainly that there is a significant decrease in the binding, certainly not as profound as in this particular case, as that one observes when someone is being treated for cancer purposes, indicating that, yes, it's very relevant and it needs to be addressed in terms of understanding potential effects that smoking have. So if you think about it, if aromatase is the one that is transforming estrogen, as when a woman goes into menopause, the production of estrogens from the ovaries goes basically, I think, back to zero. But the production of estrogen is mediated by the metabolism from testosterone into estrogen. So if a woman is a smoker, their concentrations of estrogen would be lower. And this could explain, for example, why female smokers reach menopause at an earlier age. But it also could explain, interestingly, something that is similarly paradoxical, which is if you know that if you smoke, you're at higher risk of cancers. But if you already have a cancer and you are a smoker, if you have breast cancer and you are a smoker, your outcomes are better because you may be inhibiting estrogen, just like when you give Borosol as a treatment to inhibit the production of estrogens, which actually, if you're estrogen positive, that can result in proliferation of the breast cancer. So this is an example about how you can use this to understand the actions of drugs in the human brain. Now, I'm not going to let you go so simply because, again, and I was interviewed today about marijuana, and I said, reality is these drugs, when you smoke these drugs, when you smoke things, you're not just smoking and inhaling the thing you want to do, which is nicotine, or in the case of marijuana, 9-THC. You are smoking a lot of cannabinoids. When you are smoking cigarettes, you are smoking a lot of compounds. And there is a Harman compound that actually has been shown to have that you get actually in high quantities when you are smoking cigarettes. So again, we were impressed by a study that had been published in the 80s, which took rats. They took rats, then they took their plasma, and they exposed them to cigarette smoke. And the researchers published a paper saying the concentration of monoamine oxidases was inhibited by smoked cigarette. Now, why are monoamine oxidases important? We have two monoamine oxidases, monoamine oxidase A and monoamine oxidase B. And both of these monooxidases in the brain and in the body are involved with the metabolism in the brain of catecholamines, and in the body of a wide variety of amines and other toxic substances. Very, very important. They have been the targets of medication. For example, there's in Europe and in the past in the United States, one of the most frequently used antidepressant medications were drugs that were acting as inhibitors of monoamine oxidase A. In the neurology world, for example, there is a lot of interest to develop drugs that are inhibiting monoamine oxidase B. So there is a lot of interest. These are very important enzymes that regulate the concentrations of neurotransmitters. Again, using positron emission tomography, we have labeled both the monoamine oxidase B using C11 deprenyl and the monoamine oxidase A using C11 chlorgeline. And we studied then, based on that particular finding, whether there were differences in the expression of these enzymes in smokers.
I have a quick question about that probe.
Mhm.
Why is it deuterated?
We actually were interested on deuterating just to know where the reaction of the enzyme with a chemical compound occur. So by comparing the deuterated versus the non-deuterated, we could just actually figure it out. So the deuterated was to try to understand how the enzyme acted directly with your radiotracer. So we've done that, for example, we did that recently with labeling alcohol and deuterated alcohol to try, again, to extract information about the metabolic pathway.
But is it needed in the radiotracer
No,
itself?
no.
Okay.
No, this was just mechanistic in our brain to try to understand actually how it worked. And what we found out of this particular for deprenyl is an interesting, deprenyl is a suicide inhibitor. So once basically that the enzyme attaches to the deprenyl, you have to produce the enzyme de novo. So deprenyl will inhibit this enzyme for a very long period of time, which then brings to question why, when you treat therapeutically, that's the first thing that says, my God, we're giving deprenyl on a daily basis. It's an irreversible inhibitor. So why is it necessary? And if it is necessary, it means that there's another mechanism for its therapeutic action, not just inhibition of the enzyme. Because the inhibition of the enzyme is very, very long lasting.
There is another thing, guys, that what Nora was saying before, some monoamine oxidase inhibitors, particularly one, razagiline, is so far the only neuroprotective agent that we seem to have that somehow slows down some patients with Parkinson's disease. So there is a subculture within the monoamine oxidase inhibitors that is very interesting.
Yeah, no, it's very intriguing, and this had generated actually before in animal experiments. They were seeing that the animals where you inhibit this monoamine oxidase live longer, and then they try to do it, and this has generated a lot of studies because, of course, we want to stop the progression of disease, but the other issue is, could you also stop the progression of the aging of the human brain? So I guess, so you do believe that it slows the progression of Parkinson's?
It does in some patients, yeah.
Okay.
And we have no idea why.
So I was asking Walter the other day, and he said, well, what do you think? And I thought, because I say, and so I didn't realize that there's more of a consensus that was, and send me the paper because I should know. I mean, my sense was that it was very unclear. But your sense is that in some patients it really does slow the progression.
It does. That's why Phasic is the commercial name. Razagiline is the first choice in Parkinsonian patients. Actually, the first treatment that I use in Italy, for example, before the other monoamine oxidase inhibitors, because in some patients it just slows it down.
Is it a reversible? That should be easy to figure out.
When you say it's reversible, you mean does it?
Irreversibly bind, or does it bound and then detach?
It's a reversible.
And the pharmaceutical, one of the aims has been to develop compounds that are not irreversible inhibitors, because it gives you much more flexibility with the pharmacokinetics.
I think it's reversible. Religion is reversible, I think.
Excuse me?
If I remember correctly, it's reversible. The razagiline.
Okay. Yeah, that's probably. But Anto is Googling it. The answer will come from Google. When in doubt, Google. And so says, when in doubt, I'm going for it.
I will be there with them.
But so here you have
Irreversible, yes.
irreversible.
Yes.
Reversible or irreversible?
Irreversible.
Irreversible.
Yeah.
Okay, so here you have the study that we did with the monoamine oxidase B. The study for monoamine oxidase A is the same, but I'm not showing it. So here you have the non-smokers, here is the smokers, this is the concentration, and these are, one of the things that was very, very relevant was to rule out the possibility that this was genetically driven. Now, this was non-trivial because there was a whole literature out there, and I almost applied for a grant to do this, to actually, that believe that one of the risk factors for alcoholism had to do with the fact that you have low concentration of monoamine oxidase. And I remember that I had started to write a grant and all of that when we got these results, and I said, it makes absolutely no sense. And it was based on the findings that when patients that were alcoholics died and they measured their brain, there was very low concentrations of monoamine oxidase B. But if you think about it, alcoholics, 80 to 90% of them smoke. So that's when I said, aha, don't go by the wrong route. So we needed to actually, so we recruited patients who had been smokers but who had not smoked for 2 years. And you see these patients that were former smokers do not differ at all from the control. So this is not genetic. This is the effect of the smoke. And we took monkeys. We actually gave them nicotine. Nicotine is not doing that. Nicotine does not block monoamine oxidases. This is a Harman compound that is in smoke. It's actually, we also did monkeys where we exposed them to the smoke, and then we measured how long it took for the enzyme to go back, and it was approximately 3 months before the enzyme was fully restored. So this is a long-lasting effect. And so when you have data like this, you then stand back and say, well, what is its significance clinically? Monoamine oxidase A inhibitors are used as antidepressants. And one of the most frequent complaints that patients that are smokers have when they stop smoking is they feel very depressed. Moreover, if a patient has a past history of depression and they stop smoking, they frequently relapse. And this is, of course, clinically very relevant because if you want, and you want to make your patients that have depression stop smoking, you have to be aware that smoking, through its effects, both nicotine has antidepressant properties, but apart from that, through the inhibition of the enzymes, cigarette smoking has antidepressant properties. So if you're going to have a patient that's depressed, want to stop smoking, you have to think of something that will buffer those pharmacological effects of the cigarette. And now there are clinical trials combining, for example, nicotine with monoamine oxidase A and predominantly monoamine oxidase B inhibitors. Managing monoamine oxidase A inhibitors is not simple clinically. That's why they are not used so frequently, and with some potentially interesting therapeutic effects. So this is, again, how understanding, going beyond what we just focus nicotine receptors, we're now finding that nicotine has effects directly on enzymes. I demonstrated it for aromatases. And also that the cigarettes is also influencing one of the most important enzymes vis-a-vis the regulation of catecholamines in our brain. But these enzymes are also in our body, and then again, this is very relevant for toxicity. So here you have the same study using monoamine oxidase B, ligand deprenyl, on a non-smoker, again, positron emission tomography. This is a planar image, all the whole body. This is the head, this is the lower parts. You see the brain, very high uptake. You see here, you have the heart, the lungs. You can see a shade. You don't have strong images of the lungs because the lungs is predominantly, you think about them, they are chambers, so the epithelia are very, very thin. And that's why you see them like a shade, but not strong like the kidneys, very high concentrations. The liver has very high uptake, and the heart. Now look at the smoker. So you can see that the effects are not just on the brain. The guy may want the nicotine just to go to the brain, but it's not just. I mean, you see absolutely nothing in the lungs. The lungs become completely dark. The heart is very, very, very attenuated. The kidneys, you see the attenuation. And this is where actually because the level is so low that you can actually start to visualize the place where you injected your radiotracer, which you don't see in the other one. So there's a very significant reduction in the concentration of these enzymes across all of the organs. And this highlights why smoking cigarettes actually is not just associated with cancer of the lungs, but cancer of a wide variety of cancers. These are some of the cancers that are increased, the use of which is significantly increased by smoking cigarettes. But it goes beyond that. Smokers have dysfunction of multiple organs. And this is likely, in part, reflects the fact that by inhibiting one two of the enzymes that are involved in detoxification from toxic compounds, you of course are making all of these organs much more vulnerable to toxic effects, which in turn could also be one of the mechanisms that facilitates the component that facilitates cancers of other types of tissues, facilitating them. So drugs, whether we like it or not, are going to the whole body. Now this is also very relevant, and I already hinted it to you in pregnancy. And we know a lot about the deleterious effects of alcohol drinking when someone is pregnant, and is the #1 cause of mental retardation in the United States. But there's much less attention to the effects of nicotine during pregnancy. They are not as severe as alcohol, but they are actually quite severe. I guess that we tend to equate what we see in adults into the fetal development. So in NIDA, for example, we've been funding millions of dollars of the deleterious effects of cocaine exposure into the fetus, methamphetamine exposure into the fetus, and they were calling the crack babies. And I remember looking at the literature, and the concept was that the effects on these children were much worse with nicotine than with cocaine. And this was counterintuitive. And I said, well, either you believe the data or you at least don't even bother to collect the data if you're going to say, no, I don't want to believe it. But nicotine seems to have a more deleterious effects. So using this type of technology, for example, you can do something that is extremely difficult to actually measure the pharmacokinetic of drugs in the fetus, in vivo, non-invasively. You cannot do this in humans because it's radioactivity and you cannot give radioactivity to a fetus, but this is a non-human primate. What you're measuring here is C11 labeled nicotine, so it allows you to look at the distribution of nicotine, very high uptake in the brain. This is the stomach with the baby. So here is the placenta, this is the liver, this is the head. And you can see very, very high uptake of nicotine into the placenta, which actually is likely to underlie some of the toxic effects of nicotine. But you also see very high uptake of nicotine in the liver. So you know that a very significant amount of nicotine is getting into the fetus. And then you see the brain. Much lower than the female brain, but you actually, what's very interesting is when you look at the kinetics. So here you have the fetal, the mother, and this is the fetus. Even though it's much lower in the fetus than in the mother, the clearance is much lower. So what you are doing is exposing the fetus for a much longer period of time than it is with the mother. So we left it at the notion of the pregnant monkey, that you can see the uptake of nicotine actually inside the brain. And these technologies are really very flexible in understanding fetal effects because, for example, this is a study that was led by Elena Benveniste. But then subsequently to this study, she had another one where instead of using C11 nicotine, she actually used FDG. And you remember FDG allows you to look at glucose metabolism. So she studied glucose metabolism in the brain of the fetus before and after cocaine. And she showed that cocaine in the fetus brain affects brain glucose metabolism, just like it affects it in the mother's brain. So you can go from inquiring vis-a-vis the distribution and uptake of the drug itself to actually assessing the pharmacological effects directly of that particular drug in the fetal brain. It's obviously a technology that is complex because you have to do these studies in animals that are pregnant, you have to anesthetize and you have to give them drugs. But it's actually a very powerful tool. Now this is not a study from our laboratory, but I wanted to show it to you just to get you another perspective about how these type of studies can then inform public policy. Because what you want knowledge is to be able to influence the way that we make regulations. There's been a lot of interest in the public health community to understand the potential adverse effects of secondhand smoke. And the Institute of Medicine at the National Academy of Science was charged for doing a study to evaluate the strength of the evidence that secondhand smoke was deleterious. And they meet several times during 1 year, some of the luminaries of the field. And then they come up with a report and they say, for pulmonary diseases, the evidence is very, very clear that secondary smoke is actually, what you sort of call it, secondhand smoke is very deleterious. it actually enhances the risk of asthma, for example. And so they go like that for each one of the tissues. And in the brain, they say the evidence is actually quite poor, but there is some evidence that secondhand smoke may increase the vulnerability for addiction later on. There is some evidence, for example, that secondhand smoke may increase the vulnerability of obesity of the offsprings. And I just gave you data to show that in the case of aromatase, that if it is inhibiting the aromatase in the fetus, which is likely to be doing it, that in turn may be influences behavioral patterns. So this is a study that looks at the consequences using positron emission tomography of secondhand smoke to see to what extent nicotine from secondhand smoke is delivered into the brain. Here is the studies at the baseline condition. This is a ligand that binds to the alpha-4 beta-2 nicotine receptor, which is one of the main targeted receptors for the rewarding effects of nicotine. And they studied smokers and non-smokers. The findings were exactly the same. And they studied them on two conditions. One of them is in a room like this one, and then they took them and exposed them for 50 minutes in a room that was full of smoke. And then they repeated the PET scan. And this is the images at the baseline, where they are in a room like this one, where no one is smoking. And these are the images that they obtained when they were 50 minutes in a room full of smoke. And you can clearly see the decreases in the binding. Why? Because nicotine is occupying the alpha-4 beta-2 receptor, which means what? That nicotine is being delivered at significant concentrations into the human brain by secondhand. And then they went on and measured, actually they did it differently for the smokers and the non-smokers. The findings are the same on different areas of the brain. So this is the baseline. This is secondhand smoke in the thalamus, brainstem, cerebellum. So across all of the brain regions, there was approximately a 22% receptor occupancy by nicotine when you get it through secondhand smoke, indicating, of course, that this has consequences. Now, let me tie your brain to the image that I just showed you, right? So, you know, I just actually showed you that nicotine is going into the fetal brain. So if someone in a pregnant woman is exposed to secondhand smoke, guess what? It is very likely that the nicotine is going to end up into the fetus. And a question that I always ask myself, because I've been very, very intrigued, I was one day reading a paper in the Journal of American Medicine, JAMA, and they were stating something that appeared to be of interest vis-a-vis one of the consequences from something that has been very positive but can have some negative effects. As you know, that the banning of smoking in public places has been very beneficial in decreasing the amount of smoking in the population. But what this article was reporting was that what they were finding was that children of parents that smoke now have higher concentrations of cotinine in the plasma after the implementation of this policy that does not allow you to smoke in public. Now, why they were saying, well, why could this be? This could be because now the parents that cannot smoke in open, in public places, were maybe smoking much more in their homes and in their cars, so that the children get exposed to higher concentrations of nicotine. Now, my brain, as sort of jumpy as it is, immediately addressed the question about, well, what are the consequences? Think about it, a child that sleeps in a house where the parents are smoking inside the house, is getting nicotine, then goes to school. He or she is going to go into withdrawal. And now we're picturing that, and how would it manifest itself? And I predict, and immediately I went to one of my colleagues that does work on ADHD, and I said, have you looked at this parameter? So why don't you start to evaluate? And I think it is relevant because understanding those dynamics could then help implement policy. So I've always very sensitive to the notion to what extent you implement policy that pertains to the personal space of an individual. Can you forbid smoking in the house of a person? And immediately I'll say, of course no. But when you have data like this one, if you have parents that have children, then could you justify a regulation that say, we really do not want to smoke if you have children in your house? And I think that data like this one basically would indicate that it is actually could be potentially deleterious for those children.
Nora, I think those studies were done in a car, actually. He had this car which he adapted as a smoke.
So it was in a car?
It was in a car. It was yeah, I remember.
I didn't know that.
One of the students did it. Yeah.
I didn't really know that, but I think it is actually quite fascinating. Now, the last two examples, one on genetics and the other one on medication development on these molecular imaging techniques to get you another perspective on it. And this is a very elegant study, a beautiful study done by Marcus Kiley at the intramural program in the Alcohol Institute, which actually was looking at the mu-opioid receptor and how the mu-opioid receptor may ultimately influence the response to drugs. Now, why this is important? Well, as you know, and I don't recall exactly the program, but I think that by now you've been exposed to these polymorphisms. Certainly, Jonathan spoke with you. Did you speak to them about the mu-opioid polymorphism?
But actually, the meta-analysis suggests that it's actually protective.
It's a protective, whichever way. We know that it's a receptor.
It's a small effect.
The mu-opioid receptor gene has actually, there is a SNP in which you basically are changing an A for the G. So this is supposedly was called, I was considering it a risk allele until now that Jonathan is telling me that it may be a protective. Bottom line, there is a polymorphism that is functional that actually modifies your sensitivity to heroin, to actually it modifies the sensitivity to endogenous opioids. And the receptors that are encoded by this variant, the 100G, bind to beta endorphin with 3-fold greater affinity. And I had thought that the 118G variant was associated with increased risk for alcoholism. But you're telling me now that it's associated with protection? Guys, look at this as an example of what happens with science. So what we think we know then is turn around on the opposite direction.
But the last one is correct, especially in males, right?
Yeah, that's the one which is correct. I think the second one has been questioned as well. Very high.
This one has also been questioned.
No.
This one. Okay. Well, I'll tell you, this is the data as of the PubMed. So guys, just sort of the lesson about it, which is actually...
Don't read.
No, Chris, I ask the same. Sometimes myself the question, I get very excited about a paper and I start planning all of these experiments and then it turns out that it's wrong. So sometimes we say, don't read, but if you don't read, you don't get inspired, Chris.
No, but it's very important to talk to a lot of people who are actually doing the experiments.
Correct. I think that this is a lesson, guys, but don't listen to him of not reading. Read. I'm going to oppose you there. Read literature.
No, but it's a large meta-analysis that Nancy Saccone has done.
Okay, so I have to start by reading it.
I will send you the pre-publication.
Okay, and based on that, I may modify my slide here. But as I was before Jonathan told me this, we know that there is a variant that is associated with alcoholism. If it is the G or the A, really of this protection, but there is an association. And then there is this variant that when you're an alcoholic and you give naltrexone, you have a better therapeutic response. So Marcus Kiley went on and actually did a series of experiments to try to understand that. And so there are two studies, one from Kiley's group and this one from Wert's group, and this is more recent, in which they were actually trying to understand whether this allele, the G allele, which supposedly has higher affinity for the endogenous opioid, which Chris was telling me that they are starting to question it, would have a different binding characteristics. So he studied controls and alcoholics. Suffice it to say that the effect of the polymorphism was the same, which led to lower binding of the ligand. So you see here with a G for both alcoholics and control. Now this is carfentanil, and this ligand is sensitive to endogenous opioids. So the interpretation was that in the G allele, which has higher affinity for the endogenous opioid, this led to competition and binding, and so the carfentanil could not bind as well. So that was the interpretation. The other very interesting finding, as you see, is that the alcoholics have higher levels of this opioid receptor binding parameter, which again was interpreted to reflect the fact that there was a decrease in endogenous opioids competing for binding to the receptor. So this was, in a way, consistent with initial reports stating that the G variant have higher affinity to the endogenous opioid. Now, what Heilig did was take it one step further and say, well, how does that influence the responses to alcohol? So what you have here is that he studied individuals that have the AA allele versus individuals that have the AG allele. He used the C11 raclopride method that I just showed you. Instead of using a stimulant drug, he gave alcohol. And what he found was actually quite interesting. Only in those individuals that have the G polymorphism did alcohol actually decrease raclopride binding, which is to be interpreted to indicate that only on those individuals with a G allele, alcohol was able to significantly increase dopamine. And he actually replicated exactly the same findings on a transgenic mice in which actually had been, that expressed the same mu-opioid variant, indicating in the rodent studies that the G variant was much more sensitive to the ability of alcohol to increase dopamine, which evidently will be brought about by the regulation of mu-opioid receptors of dopamine self-firing and dopamine release. So I guess this is interesting because we are extremely interested on understanding how do genetic genes that are associated with risk or protection. So again, I don't know if this is risk or protection. It clearly is showing that this polymorphism is linked with a differential response in the dopaminergic system to a challenge with alcohol. But it does, so when you are finding a gene that is associated with disease, this type of technology can allow you to inquire the system and extract that information.
But Nora, isn't it true that a lot of people are very sensitive to drugs, the drug effects, are less liable to get addiction problems? I mean, I think that's been shown in a number of cases. So if you need a lot of drug to get the effect, then you become more susceptible to the addiction process.
And with Shockett, that has been actually a perfect example, his work showing that the individuals that have very low behavioral responses to alcohol are at much greater risk for alcoholism.
So that proves it?
Yeah, no, I'm just basically, again, I do not, that's why I say to me at this point is not telling which one may be a greater or lesser risk. But I would tend to agree with your statement that having a more robust dopaminergic response to alcohol in my brain would mean that you have a protective effect. So I'm not disagreeing with you, but since this in his interpretation was different, but that's why I'm just my point on this one is that here you have a polymorphism that is influencing the response of the dopaminergic system to alcohol challenge. That's what is so fascinating in my brain. Whether this is protective or increases your risk, I think that has to be determined. Now another very important example and a lot of interest, so in the psychiatry literature and in drug addiction, there's been certain genes that have capitalized a lot of attention that are common across a wide variety of psychiatric diseases. So the mu-opioid gene actually has generated a lot of interest, not just in drug addiction, but also has started to generate interest in other conditions like chronic pain. One of the other genes that has created a lot of interest is the genes for monoamine oxidases. And I was sort of telling you why these are so important. And I guess that this probably was one of the first genes that in which they found what the equivalent of a knockout situation in humans. Because what happens with the monoamine oxidase A gene is a gene that is expressed, that is located in the X chromosome. So if you're a male and there was a family where they actually, one of the X chromosomes did not express this gene, that male will be a knockout. And this is a very, very rare event, and they wrote a paper about it, and then someone wrote a book about it, because the family of these individuals had a history where all of the males had a history of violent behavior and ended up in jail. And so that generated the question of if you don't have this gene, you're actually generating a prototype that is more violent. And so that led to animal experiments where they did knockouts, and they actually do show in certain backgrounds that these result in an aggressive phenotype, but not in other backgrounds. And in humans, it led to this really interesting paper by Caspi published in Science that was very, very controversial, but then has been replicated several times, in which they were looking at individuals on the basis of a polymorphism for the low, what they call the low monoamine oxidase A, which would be associated, they claim, with low expression in the brain, because there had been some in vitro experiments that suggested so, versus those that have the high allele, which was associated, according to this in vitro experiment, with high concentrations. And they actually determined to what extent this polymorphism influenced the prevalence of antisocial behavior in these individuals. And what they found was an interaction with the past experience of the subjects. Those individuals, so they look at those that have vis-a-vis exposed to physical maltreatment, whether have known, probable, or severe. And what they found was if you have the allele that leads to supposedly high concentrations of monoamine oxidase, you did have a slight risk if you have severe maltreatment, but it was very relatively low. In contrary, what happens was those individuals would have the allele that's associated with low expression. If they have no childhood maltreatment, they don't have this antisocial phenotype, but if they have severe maltreatment, there you saw the emergence of the behavioral phenotype. And this was, I think, probably the first gene by environment interaction that was published in the literature. And again, this has been replicated, and it has opened up a whole series of questions, including researchers then started to go back and forth and seeing how does this affect brain function, and is it related in any way to actually areas of the brain that are involved with aggressive behaviors. Many papers saying yes, if you have the high versus the low monoamine oxidase allele, that's going to result in differences in brain structure as well as brain function. This is one of the studies that were done to assess differences in brain function, and what they showed is the polymorphism that was associated with a low concentration supposedly leads to much thinner orbital frontal cortical areas. And they also saw changes in the amygdala. Others have seen changes in the amygdala. This is a study that we did at Brookhaven National Laboratory using a very simple paradigm, which was challenging subjects with functional MRI with the word no, emphatic no. And this idea came to me when I came to the government. I mean, I'm being very political. No, I swear this is the truth. I'm not making it up. I came to the government and said, no, Nora, you cannot do this. No, Nora, you cannot do this. No. And so one day I was running and I said, this thing of the no, it must be harming my brain. So I came to the lab and I said to one of my postdocs, can you do an experiment to see what no does to your brain? And that was, and she said, yes. She said, yes.
She said no.
No, she said, yes. She did this is Nellie Klein, did a very, very clever experiment and showed indeed that when you activate no, you actually activate inhibitory pathways. It's a very profound effect. But then she very cleverly said, Nora, let's try this paradigm for patients that are aggressive versus non-aggressive. And then she said, well, Nora, let's look at the polymorphism for the monoamine oxidase A. And she did indeed find differences in the activation of the dorsolateral prefrontal cortex to the no between those that have the high and the low. And everything was very nice and dandy and sort of says, yes, high concentrations of monoamine oxidase are actually very good for you, but if you have low concentrations of monoamine oxidase are making you aggressive. And my brain was saying, that doesn't make any sense at all. Because if it were true, when you're treating your patients with an antidepressant that inhibits monoamine oxidase A, they should be aggressive. And they are not aggressive. And all of these cigarette smoking people should be aggressive, and they are not. Because you are inhibiting the enzyme. So if it were so simple as low monoamine oxidase is resulting in low concentrations that drives these, then 2 + 2 are not 4. I mean, you sort of actually, as a clinician, patients don't become more aggressive on these antidepressants, nor people that smoke become more aggressive. It's more like they become more aggressive when they are irritable and in withdrawal. But anyway, so we started to wonder and says, well, we have the tools. Let's look at it. Let's look in the brain of people that have the high and the low polymorphism, regardless of what everybody's saying, and let's measure it. And this is the data. Concentration of monoamine oxidase A, individuals with a high polymorphism, individuals with a low polymorphism. These are the average images, identical concentrations. So then how do you bring these two together? And I think that this is one of the challenges that we see in psychiatry. So we're identifying a gene that is associated with a behavior. But what we don't know is how does that gene result in that behavior. And in the case of the brain that takes such a long period of time to develop, one of the effects may be during the actions on the fetal development of the brain.
Sorry, Nora, I missed this. The bottom of the two groups, from a behavioral personality perspective, and the top were absolutely identical to each other.
So you
there
mean to
was-
say these with concentration and these?
There you go, yes. The
No,
two-
they are not.
They are similar in terms of personality.
No. And so then we went on to actually look at the characteristics, and I'm not showing you the data, but we published that individuals, whether you have the high or the low, that tend to have very low concentrations are more aggressive.
Oh,
And there's a very nice correlation between the concentration of the enzyme in the brain and aggressiveness. But it's not mediated in the context of actual concentration by the gene.
okay.
So the gene, so what then led in my brain to say, well, what's probably happening is that it's likely to be a developmental effect. If you don't have that gene, that effect is going to start from the beginning. So it may then result to these changes in brain function that we are observing with these other types of tools. So it's not that they don't differ, but that the effect is not driven right now. And so what you say is, well, why is it that you have no differences at this point? And again, it's like anything else. It's a gene and environment. And I just showed you an environmental intervention that leads to differences in concentrations of the enzyme, smoking. And that's very profound. So it's likely that other factors may also be influencing the concentration of the enzyme. So it's not just the genes that are actually responsible in an adult for what are the levels of the enzyme.
So has anybody identified epigenetic changes?
Yes, we've looked at that also, and indeed, absolutely. There is the epigenetic marks predict the concentration of, and that's Elena Shumai in our group that did that study.
And I think there's also stuff by Saloni on looking at interaction between high and low levels of serotonin and parenting.
Yeah, there is, absolutely. You know, that's correct. But I mean, I chose this example because I actually wanted you to realize as we go into the whole field of genetic in brain diseases, particularly mental illness, including addiction, we have to recognize that the associations are going to be complex, and the effect of a gene may be limited to a particular window during brain development. So it's not that we are going to necessarily see a one-to-one correspondence in the present. And this is a diagram that Ruben Baller at NIDA did to actually, he tried to illustrate it diagrammatically. So you have a genetic load, and that leads to a brain outcome. But those brain outcomes are going to be presented themselves in many ways that will affect, for example, personality characteristics that then lead you to take drugs, and then that leads to the process by which you get addiction. The way that these genes are involved in brain development. These genes, many of them, the ones that are identified as relevant, are also modulating and mediating our sensitivity to stress, which, as you know, in turn is one of the factors that increases your vulnerability to addiction. The extent to which these genes, for example, very simply, the question that you brought up, your sensitivity to the drug. So if you are very tolerant in certain instances, guess what? You're at greater risk. If you're very sensitive or you cannot metabolize, you are protected, like with the case with alcohol dehydrogenases. By the way that genes affect neuroplasticity, and therefore the changes that occur with repeated administrations. And ultimately, a very important aspect of all of this is the role that these genes are going to be playing in our tailoring of medications for treatment of these disorders. My final slides before I have to run into the airport, and there is my mind, he's actually over there, is the concept of medication development. And so let me actually jump these studies from us and concentrate on a study that's not from us, because it's very, very exciting in the whole area of medication development. And it was very embarrassing. There was actually, guys, you know, one of the priorities of the Institute has been to develop medications and treatment interventions. and I won't have time to answer those questions because I'm going to have to leave. But to me, I basically believe that we need to develop strategies that can help us improve the outcomes in people that are addicted, which actually we have very few medications. And one of the interesting strategies is vaccine development. And so we are investing, we're promoting researchers that come from the vaccine arena as well as behavioral scientists to work together in order to develop medications for nicotine, for cocaine, for heroin. Those are the ones. And actually, also we've been trying to work with vaccines or antibodies for methamphetamine. And this is an example for a cocaine vaccine. In fact, these are studies done by Crystal, and he has been working in collaboration with George Coop, who came and gave a presentation here. The idea, I don't know, if did George speak about vaccines?
Yes, a little bit. Why? I voted. He didn't go into a lot of detail about it, put it like that.
Well,
He did mention it.
Ron Crystal actually came to me. I mean, basically, he said, Nora, I want to speak with you. I says, okay, come on, speak with me. And he said, Nora, you know, I have developed these, he was developing vaccines for, I don't know what, bacterial or nosocomial infection or something like that. And he says, but you know, I have very strong antigenic responses. And what I work is, I work with an adenovirus, and I basically put my antigens in the fibers of the virus, and that really creates a very strong antigenic property. Would you be interested on that? And I said, let's speak about it. Can you put nicotine or cocaine? And he says, yes, we can. And that's why we sort of started to fund him, and he had been developing some extremely interesting probes and working with George Coop to test them. So they basically link a cocaine analog to these fibers. They generate a vaccine, they vaccinate the animals, and then what happens is that the antibodies that are formed in response to the antigen will bind to the cocaine, interfere with its entrance into the brain. So you have, and there are many challenges with vaccine development. You want to have high antigenicity so that the antibodies are very high. You also want to have specificity because you don't want antibodies that go for something that is not cocaine. And so those are non-trivial phenomena. And also you want the antibodies to have a long-lasting effect. This vaccine this one particularly has very, very good properties. So this is in monkeys, in which actually they are measuring the occupation of cocaine of dopamine transporters. Cocaine binds to the dopamine transporter. And the ligand that they are using is C11PE2I. And this is a baboon brain, and you have the two axial planes in the basal ganglia. That's dopamine transporters are available, so the ligand can bind. This is a sagittal plane. They give the animal cocaine, 1 milligram per kilogram IV, which is basically on the high dose, but it's a regular dose used by cocaine abusers, and you can clearly see that the binding goes down. Why? Because cocaine is blocking the transporter, so your radioligand cannot bind. So this is the animal before they give them any vaccine. They vaccinate the animals, and then 10 weeks later, they do the injection, the challenge of cocaine again. So this is before they give the challenge, and you see basically the nice binding. This is the axial plane, this is a coronal, this is a sagittal, binding dopamine transporters. And this is, they give 1 milligram per kilogram of cocaine, and you see basically there's no difference in binding. The dopamine transporters are still available because cocaine never got into the brain to bind to them.
So Nora, did anyone do studies where they measured self-administration before and after the vaccine to see whether you have similar effects to the antagonistic effects of, let's say, a dopamine given?
They have done those experiments both for cocaine, they have done them for heroin, and they have done them for nicotine. And it does interfere with self-administration, it does interfere with condition-place preference, and in the case, this is data, again, the heroin data is from George Coop laboratory, it interferes with the analgesic effects of heroin. So it is, but the problem is, I always say it's very easy to treat rats and mice, guys. So these very nice responses that we're getting in rodents, when as of now, the studies that have been done in the clinic, 2 large studies, one in cocaine, one in nicotine, have not been sufficiently strong to merit the FDA to say, well, we're going to approve it. So there is a signal in those individuals that get antibodies, but there are very few that develop antibodies at titers that are high enough.